Welcome to the second screencast on hormones. This screencast deals particularly with the endocrine function of the pancreas. So this first slide here requires you to know about the structure and histology of the pancreas and outline its role as an endocrine and an exocrine gland. Okay, so just dealing first of all just to orientate yourself about where the pancreas is, etc. So the pancreas here lies just below the stomach in the left side of the upper abdomen, and it's dealing with its exocrine function. You remember from GCSE the pancreas produces enzymes such as lipase, amylase and proteases. These enzymes are produced by cells which cluster around the pancreatic ducts. Okay, and the pancreatic ducts will feed into a main pancreatic duct, which then passes in the secretions of these cells into the small intestine here. Now, if I was to look more closely at one of these cells are arranged around the pancreatic ducts, and these form what are called pancreatic acini, which are little clusters of cells which cluster around the pancreatic ducts. They produce proteins, enzymes, etc., which then they release into this region here, which then gets funneled into the pancreatic ducts, and then from the pancreatic ducts, moving over here, it moves down through the pancreatic ducts, through here, and eventually into the small intestine. And forms the exocrine part of what the pancreas does. Now, if I was to look at a cross-section of these cells, if I can imagine all the cells that lie between all these pancreatic acini in this pancreas here, what I find every now and again, in amongst all these pancreatic acini, which are clustered around ducts, is I find these structures here, which are clearly cells which have different functions, and these structures here are known as islet of Langerhans. And the islet of Langerhans are the regions of the pancreas that perform the endocrine function of the pancreas, and the two hormones we're particularly interested in are the production of insulin, which lowers blood glucose, and the production of glucagon, which acts to raise blood glucose. And we'll look at the secretion of these in a lot more detail, as well as how these particular hormones work in target cells. So if I quickly go back to my slide here, this diagram up here shows much the same. Here I have an islet of Langerhans surrounded by these pancreatic acini cells, which are the exocrine function of the pancreas, producing enzymes, etc., which are released into ducts, and the islet of Langerhans here. Now, the islet of Langerhans, as far as we're concerned, has two principal types of cell. It's got beta cells, and these are specifically for secreting the hormone insulin, and it's got alpha cells, which are specifically for secreting the hormone glucagon. So, endocrine function. The endocrine part of the pancreas is called the islet of Langerhans, and these secrete cells, alpha cells, I should say alpha there, alpha cells that secrete glucagon, and beta cells that secrete insulin. And as any endocrine gland, these hormones are secreted straight into the bloodstream. Whereas the exocrine function are these structures around the outside of the islet of Langerhans, and they're known as pancreatic acini, and they're producing enzymes, and they're clustered around ducts, pancreatic ducts, which all funnel into the main pancreatic duct, which takes the enzymes and various secretions to the small intestine. And these enzymes, amylase, which is a starch digesting enzyme, breaks the starch into maltose, trypsin, which is a protease, which breaks down protein molecules, and lipase, which breaks down fats into fatty acids and glycerol. As well as those, it also reduces sodium bicarbonate, which is an alkaline substance which is there to neutralize stomach acidity. So moving on, Here is a picture which shows the histology of the pancreas. So obviously we looked at all this diagrammatically above, and as you know, when you actually look through a microscope, nothing ever quite seems as it does in a diagram. So trying to make sense of all of this, so all of these cells around here, which are these yellow things are pointing to, are what we call pancreatic acini. So there's a nice pancreatic acini. You can see the cells clustering around a little hole in the middle, which would be the cross-section of some sort of pancreatic duct. Also, these structures here would be a network of pancreatic ducts which all feed into larger pancreatic ducts. And this here may even be a larger pancreatic duct here, not so sure, but it could be. So all of these are feeding into these ducts, so all the molecules, enzymes, etc. are being released by all these cells, and they're all moving, migrating towards the larger pancreatic ducts, and eventually all of those substances get released into the small intestine. However, in amongst all of these pancreatic acini, we see these structures, okay, which are clearly very different, and these are the islet of Langerhans. Okay, and these are the structures which contain our beta cells, which release insulin, and our alpha cells that release glucagon. I'll just show you a short animation which illustrates some of that.
The pancreas is an important organ in the digestive system. It is located in the upper abdomen, directly behind the stomach and next to the small intestine. In most adults, it is between 6 and 10 inches long and 2 inches wide. The pancreas is comprised primarily of a network of tubes or pancreatic ducts that release liquids into the upper portion of the small intestine, called the duodenum. Anatomically, the pancreas is divided into 3 regions: the head, the body, and the tail. The pancreas has 2 functions: endocrine and exocrine. Its endocrine function is to produce the chemicals or hormones that regulate blood sugar, such as insulin. Its exocrine function is to produce enzymes that help to digest food. Pancreatic amylase breaks down carbohydrates or starches into glucose. Proteases break down protein into amino acids, and lipases break down fats. Because these digestive enzymes are so powerful, they are wrapped in a protective layer while they are in the pancreas. To reach the gastrointestinal tract, the digestive enzymes travel through the pancreatic ducts and are eventually released into the duodenum at the major papilla, also known as the ampulla of Vater. Once they are completely out of the pancreas, the protective layer is removed and the enzymes become active. Bile from the gallbladder also enters the duodenum at the major papilla. Bile breaks apart fat into smaller fat droplets, which are easier for lipase to digest. When the pancreas is healthy, it contributes to a healthy digestive system. However, when our pancreas does not function correctly and is unwell, we can have trouble digesting food properly or maintaining our blood sugar in a healthy range. The consequences of an unwell pancreas may include diarrhea, bloating, flatulence, oily and foul-smelling stool, weight loss, malnutrition, poor blood sugar control, and diabetes. Heavy alcohol consumption, high-fat diets, eating large meals, being overweight, and tobacco products can put stress on your pancreas, causing it to work less well. There are also some genetic conditions that affect the pancreas, notably cystic fibrosis. To help keep your pancreas healthy, eat a varied diet that is rich in fruits and vegetables. Include fish and white meats as favored sources of protein. Limit high-fat foods and alcohol to special occasions. Maintain a healthy weight, and watch your cholesterol and triglyceride levels.
So in summary, what you should be able to get from this slide, you should know the difference between the endocrine function of the pancreas and the exocrine function. In terms of the endocrine function, you should know of the fact that alpha cells, I'll just write a little alpha in there, secrete glucagon, and beta cells, just write a little beta in there, beta cells secrete insulin. You should also be able to understand diagrams such as this diagram here, with its islet of Langerhans here, and these pancreatic acinar cells around the outside. Similar diagram we've got here, islet of Langerhans with the acinar exocrine cells around, with the islet of Langerhans endocrine cells here, but also it requires you to be able to know the histology, so be able to interpret pictures like this, which shows all the exocrine acinar cells, which make up the bulk of the pancreas, but interspersed amongst all of these are these structures here, the islet of Langerhans with its alpha and beta cells. So moving on to the next slide. So this second slide looks at how insulin is able to control blood glucose, but also how glucagon can also control blood glucose. This is another homeostatic mechanism. We keep blood glucose constant by the process of negative feedback. The idea that if blood glucose levels rise, we detect that and we respond to it by reducing blood glucose levels back down to the norm. Conversely, if blood glucose levels drop, we detect that and respond by raising blood glucose back to the norm. Okay, so that's another negative feedback control mechanism which allows homeostasis to occur, i.e. keeping the blood glucose, in this case, level in the blood constant. This is all done in the islet of Langerhans, and we'll see here, interestingly enough, that the islet of Langerhans does the actual detecting. We have the sensory cell, the islet of Langerhans can sense blood glucose levels altering, and they also respond by releasing the hormones which then move in the bloodstream and influence target cells, which are the effectors, if you like, which result in changes in blood glucose. Okay, so first of all, dealing with what happens when blood glucose is too high. First of all, this is, as I said, this is detected by the cells in the islet of Langerhans, which actually do the secreting of the insulin as well, and those cells they're called beta cells. I'll just draw a little beta in there. Okay, so it's detected by the beta cells, okay, and we'll see how that detection occurs on the next slide. But for the purpose of this slide, we're just going to look to see how the insulin can cause a reduction in blood glucose levels. So the insulin is released into the bloodstream, where it acts as a first messenger, and it binds to target cell membranes, receptors on target cell membranes, and that causes the production of a second messenger within inside the cell. Okay, and those second messengers do two things. First, what they do is they insert glucose transport molecules in the cell surface membranes. Okay, and secondly, they activate enzymes which convert glucose into glycogen. Okay, which is a process known as glycogenesis. Okay, so if I just look at that in a little bit more detail with some diagrams down below. Here we have our liver cell. Also, this could be a muscle cell or a fat cell, but the purposes of this slide we're interested in the liver cell. So the liver cell on the cell surface has a membrane which is complementary in shape to insulin. Okay, and insulin can bind to that insulin receptor, and then what happens is a second messenger is produced within inside the cell. This idea of signal transduction cascade, which we talked about in a previous screencast, the production of a second messenger results in a membrane-bound vesicle which contains glucose transport molecules moving to and fusing with the cell surface membrane, and then places these glucose transporters into the cell surface membrane, which allows glucose to pass into the cell by facilitated transport, and in so doing reduces the amount of glucose in the blood, because the glucose is now moving in by diffusion, facilitated transport, into the liver cells. So that's the first thing that happens. The second thing that happens is that these second messengers also activate enzymes which can convert this glucose, once it's moved into the cell, into the storage polymer glycogen. And that process is known as glycogenesis. Key word, glycogenesis, production of glycogen from glucose. Anyway, some of that is also illustrated in this animation below.
The digestive system begins to break down the food you have eaten. The carbohydrates in this food are broken down into a simple sugar called glucose. As this glucose is absorbed from your digestive system into your bloodstream, your blood sugar levels increase. Your circulatory system then carries the glucose to muscle cells throughout your body, where it is used to generate energy. And here's the important part. In order to get that glucose that's now in your bloodstream into your muscle cells, you need to have insulin present to trigger that uptake event. Insulin is a small protein hormone produced by your pancreas. As the concentration of glucose in your bloodstream rises, your pancreas senses this increase and is stimulated to release insulin into the bloodstream. The newly released insulin plays a key role in regulating the concentration of glucose in your blood, a process known as glucose homeostasis. The insulin that is now released into your bloodstream binds to the extracellular domain of receptor proteins found on the surface of liver, muscle, and fat cells. This binding triggers the autophosphorylation of the intracellular domains, which, in turn, phosphorylate a specific substrate signaling protein. This protein then phosphorylates other downhill signaling proteins, leading to an amplification of the signal at each step. This overall signaling process is known as a signal transduction cascade. One important consequence of this signal cascade is the movement of glucose transport proteins, called GLUTs, towards the cell surface. As these storage vesicles fuse with the cell membrane, the number of GLUTs present on the surface of the cells increase, allowing the glucose to enter the cell. As a result, the glucose concentration in the bloodstream decreases. The glucose is now inside the cell, where it can be metabolized to generate the energy in the form of ATP that is needed in all of your cells. And that is how insulin helps the sugar you eat turn into the muscle that you use.
So now we can look at what happens when the blood glucose is too low. So when blood glucose is too low, that's also detected by cells in the islet of Langerhans. These cells are called alpha cells, just draw a little alpha in there, and they're responsible for releasing another hormone which is called glucagon. Now glucagon is released into the bloodstream, circulates in the blood, where again it will attach to target cells. Those target cells are actually the same target cells for insulin. So liver cells and muscle cells, and particularly liver cells, and that's what we're interested here. So the glucagon will bind to a receptor. It will be obviously a different receptor to the insulin receptor because glucagon's a different molecule, therefore it's a different shape, and we'd have to have a different receptor molecule which would be complementary to that shape. So the glucagon receptor would be on the target cell, and it also is involved in one of these formation of one of these second messengers. In fact, in this case, it is the cyclic AMP second messenger that we talked about on the previous screencast. So cyclic AMP is ultimately produced inside the liver cell, and this activates enzymes to convert glycogen to glucose. Okay, and that process is known as glycogenolysis, as opposed to glycogenesis, which is the formation of glycogen from glucose. So this is the reverse of glycogenesis. Okay, so these are key words you should know: glycogenesis, and in this case, glycogenolysis. So then what happens is that the glucose, once it's produced in the cell, will then exit out of the cell by facilitated transport and act to increase blood glucose. So moving on to the next slide. This slide deals with how the beta islet of Langerhans cell actually detects an increase in blood glucose. This is quite complicated, so you'll have to, quite detailed, so you'll have to go through it carefully. Anyway, so what happens is that glucose levels increases in the bloodstream, perhaps after a meal, and this is detected by the actual beta islet of Langerhans beta cell itself, the beta islet of Langerhans cell. So I'll just write a little beta in there. So the glucose passes into the cell by facilitated transport. It passes into the beta cell by facilitated transport. Okay, so that's shown here. So glucose passing into the cell by facilitated transport. The glucose then undergoes respiration, glycolysis, Krebs cycle, etc., and ultimately produces ATP, so increases the ATP concentration in the cell. Okay, then what the ATP does is the ATP then blocks a potassium ion channel in the cell surface membrane, and that results in less potassium ions escaping from the cell. Okay, and if you can remember the work we did on nerves, you'll know that there are things called sodium-potassium pumps which are pumping potassium ions into the cell. Okay, but because they can't escape, there's going to be an accumulation of potassium ions inside the cell. This leads to a depolarization of the cell surface membrane, which opens voltage-gated calcium ion channels on the cell surface membrane, which results in calcium ions moving in to the cell by diffusion. And these calcium ions act as a second messenger inside the cell and stimulate the movement of secretory vesicles containing insulin to the cell surface membrane, where they fuse with the cell surface membrane and release the insulin molecules out of the cell by exocytosis. Okay, and the insulin is now in the tissue fluid, which then drains into the vein, the venous system, and also the lymphatic system, and gets into the circulatory system and can be circulated around the body. Okay, so it's quite complicated. I've got a very crude little homemade animation below, which will hopefully help you understand this. So, here we have a cell. So this circle here represents one of my beta islet of Langerhans cells. Okay, so as we know, what happens is that these positives, okay, represent potassium ions, okay, and the beta cell is what we call an excitable cell, much in the same way as a neuron is, and normally it has a resting potential. It's more negative inside with respect to outside. And the reason for that is that there are sodium-potassium pumps which pump potassium ions in and sodium ions out, but the potassium ions, once they're pumped in, what they do is that they leak out through potassium ion channels. So all these positive ions, having been pumped in and under normal conditions, then leak back out again, and that results in a maintenance of this idea of the cell being more positively charged outside with respect to in. Okay, so that's the situation as normal. Okay, so we have leaking of these potassium ions out. Okay, I'll just bring those potassium ions back into the cell for a second to illustrate the next bit of the story. So what happens is glucose levels increase. Okay, so along comes a glucose molecule and enters into the beta islet of Langerhans cell by facilitated transport through one of these glucose transport molecules. Okay, so a facilitated transport molecule protein in the cell surface membrane, represented by these red lines. Okay, so glucose enters in through the glucose transport molecule. It then undergoes respiration to produce ATP. Okay, and the ATP levels in the cell increase, and the ATP blocks these potassium ion channels. The problem with that is that these potassium ions can no longer escape from the cell. So what they do is they act to depolarize the cell. Okay, so no longer is it negatively charged inside, and it starts to become more and more positively charged due to the fact that these potassium ions are accumulating in the cell. They're being pumped in by sodium-potassium pumps, but they can't escape, so they're accumulating inside the cell. That depolarization of the cell surface membrane results in calcium ion channels, which are voltage gated. Okay, the depolarization opens these calcium ion channels and results in calcium ions, which are these green blobs, diffusing along the concentration gradient into the beta cell. Okay, so in come these calcium ions, these green calcium ions. Okay, and these calcium ions act as a second messenger inside the cell and result in vesicles which contain the hormone insulin. They move to the cell surface membrane and they fuse with the cell surface membrane, and the insulin is then released into the surrounding tissue fluid. Okay, so that's how an increase in glucose concentration results in the release of insulin from a beta cell. Okay, I'll just pause that there so you can take your own notes and then look at this diagram, and hopefully that makes some sense. Moving on to the next slide,